cover. We'll take a test before finals week, and then get ready for finals, and then take your final. Your final's going to be all multiple choice. There's no written. Okay, and they're all questions you've seen. They're just I take all the tests you've taken, and I took select questions from all of them, put them together as one. We'll have a couple reviews. I'll give you a grande size review packet, and it's like 4 pages or so, and we'll cover a couple sections a day. So that's what we'll do. Okay? Okay, let's get started. So, multiple choices tomorrow. Christian's absent. That's standard protocol. Yeah. When is he going to get a letter? Soon, because he's missed a lot of school.
Okay.
A lot. And Jansen's not here.
Surprise, surprise.
You know James got my 3 letters.
Yeah, I'm sure. Okay, here we go. So chapter 11 is the cell signaling stuff. Chapter 12 is the mitosis stuff. Okay, are we ready? All right, let's go. All right, stages of cell signaling. So we have a cell, mish. Okay, stage 1 is reception. Okay, stage 2 is transduction. And we'll just kind of do something like this. Okay, here's the nucleus, DNA. So stage 3 is response. Okay, that's kind of your basics, right? And then we kind of get into the details of all of those, all right? So receptor proteins. So out of the 3 stages, what is this referring to? Stage 1, right? So types of receptor proteins. All right, let's be specific with what they're called. So this one activates inside here. You have a compound called GTP. So because GTP is present, what would that receptor protein be called? That's a G protein. Okay. So that's our G protein. Let's say we have these. Those are tyrosine kinases. I'm just going to put TKs. Okay. The deal with tyrosine kinases, the giveaway to identify them, on the sides here, what are those P's? Phosphates. It gets energized, and what do? They come together, right? And the P, it's been given a P. What's the word for given a phosphorylated, right? So tyrosine kinase gets phosphorylated. Here's one. Okay, so we've got this one, and got the little door here. Okay. So we're going to call that IGs, right? Ion gated. Okay. And so that's the gate, right? And so when the gate opens, ions flood through, right? So that's our ion gated channel. Okay. What's the 4th one? No, it's not a intracellular, which intracellular literally means in the cell. So I'll make this little horseshoe looking thing. There's our intracellular. Okay. All right. So you good on how to identify transduction? I haven't got that far yet. Secondary, I'll talk about right here. I'll get to it. Okay. So that's our types of cell signaling. Good on that?
Yeah.
Okay. Then we get into types, well, receptor proteins. Then we have types of cell signaling, right? So we've got intracellular, right? Meaning the cell is activated on the inside, and then we have the outside, which is extracellular. Okay. But what I mean by this types of cell signal, local.
When they're like actually touching.
Yeah, so local means cells are touching each other, sending signals. Okay? And then the other one, which is most of this chapter, is long distance. Okay? So remember I talked about thyroid hormone? Okay? So thyroid hormone gets in your bloodstream and goes where?
Everywhere.
Everywhere, right? And so that's a long distance kind of signaling. Okay? When a teenage boy starts making testosterone, where's that testosterone go? Everywhere. What do you see visually that he's making testosterone? Your voice, right? The voice is lower. Doesn't sound like a little boy anymore. He has armpit hair. He has hairy legs. Maybe facial hair as a middle schooler. Kind of weird, but it happens. Is that Christian? Christian makes excessive amounts of testosterone.
That was in 4th grade.
Now it's estrogen.
Now it's estrogen. Yeah. So that's local and long distance. So speaking of that, so if you're making a hormone and that's in your bloodstream, does it activate every cell in your body? Yes or no? No. What do cells have to possess to be activated by a signal molecule? They have to have a receptor. Okay? So if the signal molecule looks like that, and your receptor protein on certain cells look like that, it doesn't activate it. But if it's a different cell and it has a receptor protein like that, this will activate it and make it do something, right? So like adrenaline, the fight or flight hormone, that's a long distance signal, right? The grizzly bear standing in front of you, you're on the trail, and you got to figure out what to do. How does your body react to adrenaline? Does your hair stand up? Yeah. Does your heart race? Are you sweating? Right? So, but there's other parts of your body like your kidneys, they're not really going to work. Although you could pee your pants, but that's just a different story. That has nothing to do with the hormone, right? So that's local and long distance. I beat that to death, but there you go. Okay. So here's our receptor protein. Here's this thing. What is another word for a signal molecule? Yeah, a ligand. Okay. A ligand. All right. So now let's tie that into transduction. Okay. So let me clear my screen and make a new one. All right. So here's our ligand. We did reception. Now we're going to do transduction. Okay. So if this is a G protein, let's just say that that's what we're going to talk about. We have this compound called GTP, which activates that protein. Okay. And one of the first things, if it's there, that gets activated is maybe a substance like this. What's CAM? It's a secondary messenger. Okay, so secondary messengers are activated early. Okay, they don't come along later. They're early in this whole transduction sequence. Okay, so it's not like the proteins go here, activate a secondary messenger, then you get a response. It's you activate a secondary messenger, and that activates other proteins. Okay, so we have a secondary messenger. What's another one? What's another secondary messenger? Yeah, calcium ions. Okay. And what's the third one? IP3. Okay. IP3, inositol triphosphate, okay? So these are your secondary messengers, and all they do is help send the signal. Okay, so if you have a bunch of dominoes that you need to knock down, they are helpers to knock the dominoes down, okay? All right, so we did that, we did our secondary messengers. Now we've got this thing and this thing, and there's a whole chain of them.
CDK.
Okay, we have, we'll talk about CDK specifically in the next chapter, but these are the enzymes that help send the signal. Starts with the K. Right, so these are the kinases. Okay, these are all kinases. Okay, so kinases are the enzyme that bind to, I mean, we might as well say it, right? A molecule called cyclin. Okay, and when they get together, that it's been activated, right? So how do we know it's been energized? It's got a phosphate, okay? Which then, so this one here, let's show it. This is inactive kinase, this is active kinase. How do we know it's active? It's got the phosphate. So now that this one has been activated, it's going to bind to this one, or send a signal to it. What's going to happen to that one? That's going to get activated. So now it's no longer inactive, it's active. But this one's inactive, so what's it going to do? Activate that one, right? So kinases are the proteins that help send the signal. And since these are all getting phosphorylated, this whole thing is called a phosphorylation cascade. Okay? There's our phosphorylation cascade. You get the cascade effect, mountain stream, right?
Think of like
Dishwasher soap? I don't. Okay. All right. And then you get here to the nucleus, right? And that's actually the next thing, but we're not ready to talk about response. Okay. So, properties of ligands. All right. So, this ligand has what property? Hydrophilic, because it can't pass through the membrane, right? If the ligand is able to pass through and bind right here, then that ligand has what property? Hydrophobic, right? So it's intracellular signal. Now, two differences here. If this ligand is hydrophilic, it can't pass through, you have to have this cascade event. If the ligand is hydrophobic, it can pass through. Does this happen now?
No.
No. Now that this has been activated, it's going to just go to the nucleus and then have a response. You don't have a cascade when it's intracellular. Got it? All right. So properties of ligands, transductions, secondary messengers. Okay, apoptosis. If a cell receives the death signal, what's going to happen? It's going to die, right? So apoptosis, the cell blebs. It blebs. It kind of bubbles, making these, they look like bubbles, but they call them blebs. Okay? And a blebbing cell is going through apoptosis. It's going to die. By the way, cancer cells, do they go through apoptosis? No, they avoid apoptosis. Right. So. Okay, and I think I already hit local and long distance. So, you good? Cell signaling stuff? All right, let's move on to chapter 12. So which of the two chapters do you suppose is going to have the most information? Definitely 12. I mean, 11 is going to have a decent amount, but 12 is going to be the most. Okay. Okay, so a chromosome. All right, so you have identical chromosomes, one from each parent, right? A chromosome is made of what's chromosome made of? DNA and proteins, right? What's the name of these proteins? Histones. Right. So chromosomes are made of DNA, proteins, all tightly wound, right? And it can exist in two forms. It can be chromatin, or it can be a chromatid or some. It doesn't really matter. Okay. All right. Purpose of mitosis. All right. So here's where I noticed when we did some of our labs, you guys got confused with mitosis and cytokinesis, right? So the whole thing is called cell division. Okay, cell division is made of two parts: division of the nucleus and division of the cytoplasm. Okay, what's division of the nucleus called? Mitosis. Cytokinesis, division of the cytoplasm, right? All right, so we're dividing the nucleus, and then why would a cell need to divide? Two things: growth and repair. Okay. So if you're asexual, you go through mitosis, right? If you're bacteria, because there's no male or female, you make more of yourself, right? Okay. But that's a bad thing because there's no variation, right? Well, that's the next stuff. That's mitosis. Okay. Types of cells involved in mitosis. What are those cells called? Somatic cells. Somatic cells are your body cells, all right? Okay, sister chromatids versus homologous. Okay, let's see, go. Okay, so looking at this right here, okay, what's a which is the picture of the homologous chromosomes? Wait, what? The left. Yeah, these are the homologous. Homo meaning they are the same, right? So this is dad's chromosome. This is mom's chromosome. They are exactly the same chromosomes. You have one from each parent, okay? That's homologous. But when before a cell goes to mitosis, what do we have to do to the chromosomes? We gotta copy them, right? And so once we copy them, they're called sister chromatids, right? So here's mom's copy, because, hers are yellow, and here's our duplicate, the other yellow. And here's dad, his is blue, and there's his duplicate, the other blue, sister chromatids. Got it? Okay. Now, once these separate, like here's dad's sister chromatids, once we go through mitosis, what's this called again since it's copied? Chromatid. Once they've separated, they're now officially called chromosomes. Right? When do you officially get chromosomes?
After the division.
After anaphase. It's anaphase. Right? They line up in meta, they separate in ana. Right? And now they're back to being chromosomes. Got it? Okay. Ploidy. You know what I mean by that? Diploid, haploid. Okay, so ploidy is just what is it? So you can be N, 2N, and that's all we need to talk about so far, but there are more. So N is called haploid, and this is diploid or diploid, however way you want to say it. Yeah, diploid or diploid? Diploid, yeah. So, which one of these represents mitosis? Diploid. Does this ever happen in mitosis? No. That's a sex cell thing, right? That's the next chapter, actually. That's Thursday. Yeah. We're gonna talk about sperm and egg and all that stuff. So, okay. So this is it. Okay. All right. All right. Good on that?
Yeah.
Okay. I know I have plenty of time, so I can take my time. So I don't feel rushed. Soaking this up?
Yeah.
Yeah. Turn the camera on, go to sleep. All right. So the life and times of a cell is called the cell cycle. Okay. So the cell cycle has the longest phase, which is called interphase. I'm just going to put I, right? This is, ooh, that was a bad bracket, is cell division, okay? Cell division is made of mitosis and cytokinesis, okay? So this is all going to be mitosis, this is cytokinesis, okay? Why did I spell it with an I? Cytokinesis. Okay, so let's just focus on interphase, okay? So interphase made of 3 parts. They are called G1, S, G2, okay? Give me another phase that's not shown, but it's there. Yeah, and where's G0? Yeah, so G0 is also here. Okay, so describe what happens in G1. It's the growth. It's the regular life and times of any cell. They always go through G1, okay? Now, if you're not going to do something for quite a while, you're going to be put in G0. What's G0 specifically called? Non-dividing state / resting state. Either one's okay, right? So give me a cell that never ever leaves G0. Nerve cells. Give me a cell that's in there and on occasion it comes out. Liver. So those are good examples of things that are, G0. So that's this part, right? So this right here is the restriction points. If a cell receives the what signal? The go, not G0, but the go signal. Now we're going to go to S. And again, in S, like I said earlier, here's your chromosomes. What are we going to do to those chromosomes? We're going to duplicate, okay? And now we have sister chromatids. But a slight technicality, during S, you don't have chromosomes, you have chromatin. So you have, these are chromatids, but exists as chromatin, right? It's the skinny spaghetti stuff, right? All of interphase is chromatin, right? Okay. So we duplicate the genetic info because what does S stand for? Synthesis. And synthesis is right here, right? It's copying the genetic info. Okay. All right, that's S. G2, the cell gets a lot bigger and we're preparing for mitosis. That's it, okay? Because if you get past this go checkpoint, you are going to divide. You can't go back because you've duplicated your DNA. Okay? There's no going back. You've started it. It's kind of like you're in line to get on the roller coaster, you're almost there, you can't go back, you have to do it. Unless you're gonna be one of those people that cross over and leave and you don't do it. Until we do that.
I don't want to push it. I don't even know.
Okay, you good on that? You ready for mitosis now? Okay. Okay, so now we get into mitosis. Do you want me to speed it up or keep going this rate?
Keep going.
I mean, I can fly if you want me to. Keep going? Okay. So now we have pro, so we leave G2, we go into prophase, okay? Some major things happen. You start to see this. So what am I doing? Chromatin condenses into chromosomes. Technicality, you can call them chromatids, whatever. They're poofing up, and you can see that, right? What is beginning to disappear? Nuclear membrane, beginning to disappear, okay? Up here, these things start to move to the poles. What are those things? Centrioles. And what starts to come out of the centrioles? Spindle fibers. So 3 major things happen. Yeah, the nucleolus disappears, but that's not a major thing, okay? So 3 things. Chromatin condenses in chromosomes. Nuclear membrane begins to disappear, centrioles move, spindle forms. Got it? That's prophase. Okay? Can I clear it? All right. So then we have next one, PM. What's PM? Prometa. So here are our centrioles, right? The nuclear envelope now is basically gone. I'm just going to show a few little marks. So now officially the nuclear membrane's gone, and our chromatids are attached to spindle fibers, and they are being pushed to the middle. Okay, it could be horizontal, it could be whatever. They're being pushed to the middle. That's what prometaphase is. Okay, the transition to metaphase. Okay, so they're being pushed to the middle, and when they get to the middle, now we are in metaphase. And I'm not going to do metaphase because you know metaphase, right? Right, they're lined up. Okay, so chromatids line up in the middle, done. That's metaphase. Okay, and then anaphase. So here's our metaphase picture. There's our centrioles. Okay. What happens right here? Spindle fiber or sister chromatids are pulled apart. So now as they are pulled apart, you now have separated. Now you officially have chromosomes, right? And let's go ahead and talk about how that works. So right here is a motorized structure, okay? Because do the spindle fibers pull? No. These are microtubules, and kinetochores have a motor in them, right? So, Brenna, can you quit talking? Thank you. This is the kinetochore. Okay, what are these red things? Motor proteins, right? So in here, these motor proteins ratchet, which makes the chromosomes move to the poles, right? What makes these go? What makes motor proteins work? Energy. Gotta have energy. ATP makes that happen, okay? So ATP makes the motor proteins go, and again, what's this whole thing called again? What's the word? Kinetochore, right, okay? All right, so then we did anaphase. Then telophase. They are at each pole, and I may as well just do it the right way here. About halfway through telophase, if it's an animal cell, it starts looking like that, right? So now we're in tele. What reappears? Nuclear membrane. What happened to the chromosomes? Turn back into chromatin, and the centrioles are back, and what's no longer coming out of them? The spindle fibers are gone. The microtubules / spindle fibers are completely eliminated, right? That finishes out the nuclear part of it, okay? But while this is going on, the cytoplasm is starting to go in and being pinched, right? So now we can go ahead and roll that into cytokinesis, right? And all you really need to know is this is an animal, so what's that? Cleavage for it, right? So we have cleavage for an animal. A plant has a cell plate. That's all you need to know, okay? Because plants have cell walls, animal cells don't. Cleavage for a cell plate, okay? All right, so centrioles are microtubules, meaning they make spindle fibers, did that, cytokinesis. Excuse me. So that's all the basic part of it. The rest is the signaling part in cancer. So, you good? Moving on. You guys locked in? Yeah? Locked in.
So locked.
You see it. All right. All right, so here we go.
Another Seriously, you make them so big. Mine are tiny.
Well, I make them big so you guys can
Mine
see.
are gross.
All right.
I still can't see.
Quit.
Yeah, you need more
Keeps
glasses.
going down.
Yeah.
Okay, so CDK. All right, CDK is one of those kinases that are involved in transduction, okay? It's an enzyme that only starts working once something is bound to it, right? So here is mitosis, this is all interphase, okay? So, right at the end of mitosis, I made it dashed because what is that? That's cyclin. So cyclin is degraded, right? But then as you go through the cell cycle, by the time you're over here, cyclin has reaccumulated and now it's back. And now that it has been built back up again, what's it going to bind to? CDK. And when cyclin is bound to CDK, that makes MPF. Now we are going to promote mitosis. Right, okay? So once these are together, we're gonna have mitosis, signals are going to be sent, everything's going to work, and then we go to mitosis. By the time mitosis is over, cyclin goes away, right? And then it reaccumulates, and now it's back, okay? So, you got that? When cyclin's present, it binds to CDK. When these two make this complex, now we're officially gonna go through mitosis, right? So. Okay. I was gonna do a football analogy, but I use that. Okay, so then we get into stuff with cancer, okay? So cancer cells. All right, regular cells, if there is a gap between them, you have a cut, there's a gap, these cells are going to send what to each other? Signals. They're called growth factors. Okay? Growth factors, that's a ligand, right? And if those ligands bind to other cells, what's that going to tell those cells to do? It's going to tell them to divide, right? And they're going to divide or continue to send signals until all of these cells are touching. That's normal, standard procedure, right? Anchorage dependence. All right, so let's throw a bloodstream in here, okay? So this is your bloodstream. Regular cells, when they get to a bloodstream, they're done, okay? Because they anchor, right? Anchor meaning stay, they're being held in place, okay? Now, cancer breaks both of these rules, right? So even though, so if we do inhibition, even though these cells are touching, what do they still send to each other? Signals, which then makes them divide to form a tumor, okay? Also, do they anchor?
No.
No, blood cells will detach, not blood cells, cancerous cells will detach and get into your bloodstream and go to other places, spreading, what's the buzzword with that? Metastasizing, right. And then you might get a growth or something on your neck or your, something like that, okay? All right, so that's my go-to example. All right, so that exemplifies cancer cells, got it? All right, so two things that can happen to genes, the genetic part of it. Regular genes are called proto-oncogenes. Regular genes are called tumor suppressor genes, okay? Proto-oncogenes are normally on, okay? If they are mutated, they get switched off and they become what? Oncogenes. Because what's a cancer doctor called? An oncologist, right? So if they aren't working right, you might have that one, okay? So that protein sends signals very fast, right? It's part of transduction, and it'll send signals to make cells divide too often, right? Okay, so those all go together. The other ones are these tumor suppressor genes. If they are turned off, you're no longer suppressing tumors. So now if you're not suppressing tumors, you're going to get tumors, right? So P53 is this one right here. So if P53 isn't working right, if the guardian's not there, what's going to happen? Mutated cells are going to divide, and that leads to cancer, tumors, stuff like that, okay? I didn't put anything on BRCA1 and BRCA2, but that's the breast cancer ones, right? If P53 is working right and it finds a problem, what's it going to do to a cell? It's going to kill it. Yeah, it's going to kill it off. And so then it's gone. Okay. Okay. Malignant and tumors. Okay, and I might as well just, because you got to make sense of it. Benign and malignant, which is the bad one? Malignant cells metastasize, right? Benign cells, they've been halted. It's just a mass of cells. So you might have a tumor, it's benign, they'll just leave it. Depends where it's at. So, yeah. Metastasis.
Eyes. What about metastasize? So I did spell it. I'm pretty sure I spelled it right.
Okay, that's it. You done? Had enough? Okay. 36 multiple guess questions. Okay, FRQ has questions about mitosis. Well, it's both. It's mitosis and cell signaling. So
That's questions about.
mitosis.
That's it.
That's it. I mean, it's just if you know this stuff, you'll know the stuff for the free response. So.
Oh, I started listening. What Andy wants to do too.
All right, we good? Okay.
Oh, well, I did that.